Hello, good evening everyone, and welcome to our international free webinar. Hope you all are well in this pandemic situation. It is hard to be stay good, but we have to be. First, I want to thank you all who already joined with us and and the new participants also. Once again, I'm Umi Salma, core volunteer and head of campus ambassador in IGP. In each webinar, we meet with new participants. That's why I want to give a little description about IGP. IGP is an institution of global professionals, so student, learner, and community research providing holistic social work and education. I'm cordially welcoming you all. Today, our topic is on aspect of life science during pandemic era with two speakers Arnava Chatterjee and Pyong Jana Goshikdar. We already completed 78 webinars successfully. Please welcome our first speaker, Jana Goshikdar. Hello, ma'am.
Hello, this is Priyanjana Ghosh and I am a botanist and an academician by profession. I have completed my graduation and post-graduation in the field of botany and I started my research career in the same field in Presidency University. Currently, I am associated in Biotechnica as a senior lecturer and course architect. Earlier, I have experience of nurturing some of my degree students and PhD aspirant minds. Thank you so much.
Hello, ma'am.
Hello. Thank you so
Hello.
much for introducing me. And good evening to all of you.
Nice to see you.
Okay, fine. Thank you so much. I'll share my screen quickly. I hope I am audible and visible.
Yes, ma'am.
Okay. So, good evening, ladies and gentlemen out here. And it's my pleasure to be amongst all of you this evening. So, first and foremost, I would like to convey my heartfelt gratitude towards this esteemed institution, that is Institute of Global Professionals. They are doing fantastic job right during this the most toughest situation we are facing, that is this pandemic era, and they are conducting these webinars and they are connecting people across the world. So I would say it's a fantastic job this institution is doing, and definitely I am thankful to Mr. Kaushik Datta, the director of this institution, for giving me an opportunity to share this platform for every one of you. So yes, let's start with our session. And I would like to say that yes, already everybody's now you got to know about me, and today's discussion will be on COVID-19 etiology and pathogenesis. All right, so let's get started, everyone. I hope everybody's ready. So just to start with, no, I can very well feel that everybody can see this blank slide, right? It's written as think positive. All right, so you must be thinking that what is happening right here, because I was about to talk regarding pathogenesis, but out of the box how this think positive signboard is coming in front of us, right? So definitely, no,
Ma'am,
we
sorry
have
to
to,
disturb.
let's begin. Yes,
Ma'am,
yes,
hello, ma'am. Sorry to disturb. Your slide is not visible
yes,
now.
yes, sorry.
Your slide is not visible. You share
Okay,
share your slide now, pal.
just a second, just a second. Now is it visible? Please confirm me.
No, no,
Not yet.
no.
Just a moment, please. Now is it?
Yeah, make it full screen.
Fine. Thank you so much. Yeah, I hope it's clear to every one of you now. So, as I started with my talk with this PPT, right? Think positive. So, you must be wondering that why I am speaking or why I have started with this. So, let's begin our session with a positive note, right? So, I prefer to begin my talk with this very famous quotation mentioned by Socrates, that the secret of change is to focus all of your energy not on fighting the old but building the new. So we know that we are definitely passing through a very rough and a tough phase. But yes, we have to excel. We have already excelled a lot, and we have to do it more in the future. So definitely we have to be positive. And yes, with another quote, that life does not get easier or more forgiving. We get stronger and resilient. That is what. It's not about life. Yes, life will be unexpected, but it's we that get stronger and resilient day by day. And yes, I can proudly say this. I myself, being a COVID survivor, it's not at all tough to bear this situation. Believe me. Okay, so let's get started finally. So I want to start with a stats, which is global COVID stats, which is still, this report is still of March 2021. So as you can see, this is the numbers. It's definitely very, very alarming. Now we have now are coming up with the second wave of this COVID. So definitely this is creating another round of panic amongst the common people. But that is what we have to control there. We should not be panicking at this point of time. Okay, we have already surpassed the first wave of this, so we are now already experienced people. All right, so definitely we will be surpassing this tough situation once again. Now, today's discussion, as it says about the pathogenesis, now definitely no, we are aware that how this COVID, where that is the most sensible place or the most sensitive place of occurrence for this virus is mainly our respiratory tract, right? But definitely we do not as such be knowing about its mechanism. How does it do it? All right, like, is it this misconception is there that this SARS-CoV is similar to that of the SARS-CoV-1 that took place in the year 2012 in Saudi Arabia, right? So the thing is, we have to, or we will be discussing about those aspects today in our discussion in this webinar. So before doing that, let's see, getting introduced with the pandemic. So yes, we know that all this started from December 2019, and it started right away from China, Wuhan. We are very well aware of this. So since the emergence of SARS-CoV-2 in December 2019, there has been an unparalleled global effort to characterize the virus and clinical course of the disease. And coronavirus 2019 caused by SARS-CoV-2, it follows a biphasic pattern of illness that likely results from combination of an early viral phase response and inflammatory second phase. So we are going to definitely talk about those aspects definitely in today's discussion. So what happens is, this particular, the virus, which is popularly called as SARS-CoV, rather, we call this version to be as SARS-CoV-2. Why we call this version to be as SARS-CoV-2? Because we know that already we had an existence of SARS-CoV-1. And yes, initially it was scientists, the researchers who used to compare the present SARS-CoV-2 with the previous SARS-CoV-1, and now common people have started comparing them. So some says it's a myth, some says no, it's real. So I would say no. There is certain similarity between the SARS-CoV-1 and SARS-CoV-2, but a whole lot of differences between SARS-CoV-1 and SARS-CoV-2 also. Okay, and coming to the clinical presentations, like most of the clinical presentations are mild. And the typical pattern of COVID-19 more resembles, no, it's like the influenza-like illness. That includes fever, cough, okay, then headache, taste, smell disturbance. And definitely, no, we will be giving or ushering a update on the emerging understanding of SARS-CoV-2 pathophysiology. Okay, now, so next we have to know about that. What do we need to know about the pandemic caused by SARS-CoV-2? Okay, this should be our prior importance area. What should we know, right? Because we are actually not aware of this SARS-CoV-2 fully. So the first thing that we have to stress upon is that SARS-CoV-2 is genetically similar to SARS-CoV-1, as we had already discussed about this in the beginning. Okay. So, but the characteristics of SARS-CoV-2, there are a whole lot of structural differences between the two also. And also their viral load kinetics. Now, what do we mean by this term, viral load kinetics? Now, definitely we know about the basic steps of viral replication, right? So definitely this viral load is nothing else. That is, we can relate this concept with the concept of viral replication. So the strategy of viral replication for SARS-CoV-2 will be completely different from SARS-CoV-1. And I'll tell you about a very interesting thing here. There is a concept which is generally being stated for any virus, which is called as RO, reproductive number. And the reproductive number for SARS-CoV-2 is huge. It's definitely, the number is really high when we compare it with SARS-CoV-1. And what does this signify, everyone here? This signifies that the spread efficiency of SARS-CoV-2 is immense. Okay. So this is a very important thing we have to know. So you can see the structure of SARS-CoV-2 here, right? Now, the common structure, we get to see a virus with a spike surrounding it. So this SARS-CoV-2 is an enveloped beta coronavirus. So what is the category? It's beta coronavirus, with a genetic sequence very similar to SARS-CoV-1, and almost a similarity of 80% is there with the previous one. And also it has some similarity, some sequence similarity with bat coronavirus, which strain is called as RTG13, and it's about 96% similar to the bat coronavirus. Okay, so you can see the viral envelope. It is coated with different proteins, as you can see. And the first group of protein is spike protein, S1 and S2. Okay, so basically we know that this S protein in a virus is generally responsible for viral binding. Okay, next we have nucleocapsid protein. Okay, now this nucleocapsid protein, this membrane protein, and this envelope protein, okay, it's what it's coming to be, everyone, men. Okay, so that is the mnemonic, how to remember. Okay, I hope you will be able to retain this, what I told you right now. So apart from spike protein, the rest of the proteins include nucleocapsid, membrane, and envelope. So it's coming up to men. So this men proteins together have a very important function, and they are required for viral particle assembly. Okay, viral particle assembly. I'm writing on the screen so that it becomes easier for you to follow. Okay, so this host cell binding and this entry are mediated by S protein, as I mentioned. And the first step in the infection is virus binding to a host cell through its target receptor. Okay, and this S protein, no, it will have certain subproteins, as we have mentioned here, that there are two subproteins of spike. All right, that is, just a moment. Okay, fine. My eraser is not working. So there are two subproteins of spike. It's S1 and S2. Okay, so this S1 and S2, this S1 subunit of the S protein, it contains the receptor binding domain that binds to the peptidase domain of the receptor protein. Okay, and here I'm going to tell you about the most important receptor domain or receptor protein for this virus is ACE2. Please note this down, everyone, if you're noting it down. So this ACE2 is called as angiotensin converting enzyme to receptor. So what is the receptor for SARS-CoV-2, everyone? It is ACE2 here. Okay. And there will be a receptor binding domain to which this particular virus will be binding to. Okay. Now, so next, I would like to give you an insight that where do we find this virus in our body? Okay. And we find this virus in our body in the respiratory tract. Okay. And this peak SARS-CoV-2 load is observed at the time of symptom onset or in the first week of illness, with subsequent decline thereafter, indicating the highest infectiousness potential just before or within the first 5 days. Now, so first and foremost, the most sensitive area of this virus is the respiratory tract. That is why the highly infected and the damaged organ or that happens post COVID, it is this generally respiratory tract. It happens. Okay, because the first site of infection, it happens in the respiratory tract. And the peak SARS-CoV-2 load, in the sense, what we mean by this load, in the sense, the highest viral density, it is observed at the time of symptom onset in the first week of illness. Yes. And gradually it declines after that. But what is the trend of this virus is the symptoms will be exhibited in the first week itself. But there are a lot of post-infection symptoms that will be staying even for a period and a span of 30 days or 20 days. Yes, it happens. Okay, like weakness, fatigueness, okay, loss of appetite, this happens. All right, now. So yes, already I have given you this idea that how this SARS-CoV-2 look like. So it's an enveloped beta coronavirus, that is we have discussed, and it has a genetic similarity with these two things. Okay, now, so directly I'll go to, okay, so this is the exact structure, and there you can see how does this exactly the virus look like. So this is your spike protein. These spikes are actually the glycoprotein part. Okay, and here you have the RNA, here you have the envelope and M protein and E protein. Okay, yes. Now it's a valid question and a very important concept here to be discussed. Why is SARS-CoV-2 more infectious than SARS-CoV-1? Okay, this is a very valid question at this point of time. Now, SARS-CoV-2 has a higher reproductive number, as we just discussed in the beginning of the session. And what do we mean by this high reproductive number R naught? It means the spreading efficiency will be definitely much more for SARS-CoV-2. So both SARS-CoV-1 and 2 preferentially interact with angiotensin enzyme 2, which is the receptor, as I told you. And SARS-CoV-2 has structural differences in its surface proteins that enable stronger binding to ACE2 receptor. Got it? So this is the basic point of difference. Now, definitely, SARS-CoV-2, on the other hand, they have a greater affinity and for the upper respiratory tract. Okay, and they have a greater affinity for conjunctiva. So they can always infect, they have always a tendency to infect the upper respiratory tract, and they can conduct the airways more easily. That is their best route, I would say. Okay, like we always think, okay, let's take up this best route to travel to our destination. So similarly, it happens for the virus too. They have their favored route. All right. And that is their favorable route of traveling is they feel good to travel through a respiratory tract, making us in trouble. Correct? So this is why it is more infectious. This is the reason. That is the reason is this SARS-CoV-2 has a much more stronger affinity towards this receptor protein, ACE2 receptor. Okay. Now, next, coming to a very important aspect is duration of infectiousness. That is really a very important thing to happen. So Now, the thing is, in the respiratory tract, the peak SARS-CoV-2 load is observed at the time of symptom onset or in the first week of illness with subsequent decline, which indicates highest infectiousness potential. Okay, before or within the 5 days of symptom onset, which we have already discussed. Now, the thing is, in SARS-CoV-1, which happened almost from now 8-9 years back in the year 2012. So in SARS-CoV-1, the highest viral load, they were detected in the upper respiratory tract. Okay, in the second week of illness. That explains its minimal contagiousness in the first week after the symptom onset. That means this SARS-CoV-1, if we want to compare, what is then the difference? SARS-CoV-1, it is showing the highest viral load in the first week itself. Okay, in the first week itself. But when it comes to SARS-CoV-2, everyone, I'm sorry, I just reverted this. Okay, apologies for that. SARS-CoV-1 shows highest viral load in the second week. Like you get the symptoms, all right, like you have your fever, it's the virus is already in your body, but it'll start manifesting the symptoms from the next week, from the second week. But for SARS-CoV-1, like the virus enters into your body today, and you will be exhibiting the symptoms from the third day of that viral entry into your body. So this is the major point of difference between these two things. Okay? And that is why the people are getting, or it's getting detected faster also, which was delayed for SARS-CoV-1. And we now already know that how it is getting detected with the help of quantitative reverse transcription polymerase chain reaction. Okay, it can detect the viral SARS-CoV-2 RNA in the upper respiratory tract for a mean of 17 days after the symptom onset. Okay, and definitely this detection of viral RNA by qRT-PCR, it definitely equate to the infectiousness and viral culture from the PCR. Okay. Now coming to the most important part is the viral replication cycle. That is of utmost important. So normally, what are the steps we generally follow for a viral replication? Okay, I just want to give you a small idea about this viral replication. The first step of viral replication is attachment. Okay, and what is this attachment? So when a viral protein, it sits over the phospholipid envelope and it interacts with its receptor, then it becomes an attachment. So you can see we have certain receptor here, ACE2, ACE2 receptor. We have TMR, PRSS2. All these are viral receptors. It's very clear from this diagram. So SARS-CoV binding to ACE2 receptor and viral entry is taking place. So this is the step 1. Coming to the step 2, can you see everyone? Release of viral genome. Actually, normally if we follow the tenure of a viral replication, right after attachment, we have penetration. Okay, we have penetration. And in this penetration, what happens? Yes, this induces the capsid protein it has got. I hope everybody is aware of the term capsid protein. So this capsid protein undergoes a conformational change right at this point of time. And next, what happens? Next is uncoating happens. Okay, these steps has not been shown here, but yes, this happens. So right after the attachment, it's penetration. And all these steps are included within the concept of penetration here. Okay, so next after penetration, it will be uncoating. So what is uncoating? Viral capsid is removed, okay, one by one. And it's degraded by the viral enzymes releasing the nucleic acid. Okay, so after this, what happens? Replication. So here, can you see everyone that from the third step, it is saying translation. So till this second step, which is shown here as release of viral genome. So what is this release of viral genome? It's actually the release of nucleic acid, everyone, which I just now told you. Okay. Next, it is viral replication. Okay, so replication involves transcription and translation. So next step will be proteolysis. Okay, then it is RNA replication with the help of replicase transcriptase complex. All right. And next and finally, we have the virion release. And that means the transcription and replication of the viral genome will be taking place. And next, we have viral protein translation. And finally, we have this viral assembly, viral maturation, and definitely viral release. At one certain point of time, the virus will be released out of our body. Now, the thing is, can you see here a lot of like this genome and subgenome RNA positive strand? And here, can you see this things has been marked in multiple colors? So what they are designating, they are the different protein components, protein parts of SARS-CoV-2. So definitely it is a simple normal virus only. Okay. So it's a normal and simple virus. So only what is the differences are there? See, viruses have the same characteristics. Like if I say humans, right? So humans, the basic characteristics of human will be common. But though every individual is different from each other. No? We have the common thing in us. We have emotions. Okay, we have anger. We have the emotion of happiness. Okay, so all these phases are there in a human being, but still we differ among ourselves from one another. So just like SARS-CoV-2, no, everybody, it seems to everyone that SARS-CoV-2 is something different because it is creating such dreadful outcomes, inferences to us. But no, it's a normal, simple virus. It was there already in our society, in this world, and among us. But it has got mutated. That is why we say this to be as a novel coronavirus. Okay, and how does it differ from other viruses? That should be clear out. We should be clearing out here in this webinar. Okay, that yes, the receptors will be different from each other. The duration of the infectiousness will be different from each other in the sense from other viruses. Okay, and definitely the composition wise, it will be somewhat different. And definitely the detection strategy can be different here. Okay, so this is very, very important. And you know that several ill or the immune compromised patients, they may have relatively prolonged virus shedding, and some patients know they have intermittent RNA shedding. So even sometimes what happens, the low level results, it close to the detection limit, may not constitute infectious viral particles. While what happening is, no, the asymptomatic individuals in the sense, those with no symptoms throughout the infection, they can also transmit the infection as it is happening. And though their relative degree of infectiousness seems, it's very limited. Okay. So that is what the problem is happening now. That is the alarming thing in today's era. That is, there are quite a lot of people who are asymptomatic, and the spread is happening most from that sect of people. Okay. Now, so coming to the next part is parameters of transmission risk. This is another very important aspect to be discussed. So transmission risk depends on certain factors, and this factors in front of you already. Number one is contact pattern. Number two is infectiousness of the host. Next is socio-economic factors. So definitely most transmission, it occurs through a very close range, okay, contact, such as 15 minutes face to face, and even within, if you're standing in a 2 meter of distance. And it spread efficiently within households through gathering of families and friends. Okay, so household secondary attack, okay, rates, definitely it ranges from 4 to 35%. Just imagine. So now can you feel the essence of the lockdown that happened last year? And again, we are aware that again the rates are alarming at this point of time. So again, we have to be conscious. No, if we are conscious, if we are definitely no, if we are protecting ourselves, no, then definitely the spread can be checked, because we are not getting conscious about this thing. Okay, so that is what we have to do. If you are coming up with certain symptoms, if you are seeing any symptoms within you, just get tested yourself and quarantine yourself if you are getting tested positive. A lot of people are there, what they're doing is out of fear, they're not undergoing any tests, and thereby the spread is taking place like that. And even you should be tested if you have come in contact with any such person who had the symptoms of a common cold, even like the fever, cough, and cold. You should be tested. And then only we can check this. All right? So, like even sleeping in the same room or being a spouse of an infected individual increases the risk of infection. So, definitely the risk of infection substantially increases in enclosed environments compared with outdoor settings. Okay. So, like the aerosol transmission can definitely still factor during the prolonged stay in crowded, poorly ventilated indoor settings also. So, we have to always keep in mind these three factors we have: contact pattern, infectiousness of the host, and socioeconomic factors. They are basically forming the parameters of transmission risk at this point of time. Okay? So, yes, COVID-19, know your risk. So, had prolonged close contact, that is what I just mentioned, with someone positive for COVID-19, traveled internationally to a country under CDC level 3, traveled domestically to an area, spent time indoors. So, that is what, if you have low symptoms, then definitely go for a self-observation. If you have medium, self-observation. If you definitely, after traveling internationally to a country, if you are feeling you have certain medium level of symptoms, please self-quarantine yourself and monitor. Even for a high symptoms also, if you're getting manifested. Okay, so this is very, very important to do this. Okay, now. So route of transmission and transmission dynamics. So that is also very important. So yes, via infected respiratory droplets, direct or indirect contact with oral mucosa and conjunctiva and gastrointestinal tracts are the best routes for its transmission. I would like to say that the role of fecal shedding in SARS-CoV-2 transmission and the extent of fomite, okay, so transmission, they also remain to be fully understood. Okay, and both SARS-CoV-1 and SARS-CoV-2, they remain viable for many days on smooth surfaces. Smooth surfaces like plastic, glass, stainless steel, all these things, all the surfaces. So be aware of these surfaces. And at lower temperature and humidity. So transferring infection from contaminated surfaces to the mucosa of eyes, nose, and mouth via unwashed hands, okay, is definitely a possible route of transmission. And this route of transmission definitely can contribute, especially in facilities with communal areas. Okay, so till now, I would say that no published reports describe about this fecal oral transmission. But yes, in SARS-CoV-1, fecal oral transmission was not considered to occur in most circumstances. Okay. But yes, definitely it remains to be seen if similar transmission can occur in case of SARS-CoV-2. Definitely we have to wait for that. So now coming up with SARS-CoV-2 pathogenesis. Very important, right? That is why we are here today. So how does viral entry and interaction with target cell happens? So first and foremost, as I told you, there should be a receptor protein, and that receptor is called as ACE2. So SARS-CoV-2, it binds to ACE2, and this is the host target cell receptor. And the active replication and release of the virus in the lung cells take place, and that lead to non-specific symptoms. So what do we mean by non-specific symptoms? They are misleading symptoms, everyone. Okay? Misleading symptoms is, yes, we often get confused with the symptoms with our common cold symptoms, right? Such as fever, headache, respiratory symptoms. Okay? And definitely, there was an experimental model called hamster model. Okay, and I just like to mention here a small thing. You'll get a lot of research papers and researches that has happened with a lot of animal models. And one of the most important animal model is rodents and mouse model. A lot of experiments is going on with the mouse models. Not only mouse models, with monkeys, with apes also. All right. So yes, coming back to the topic. So in an experimental hamster model, the virus causes transient damage to the cells in the olfactory epithelium, leading to olfactory dysfunction that may explain temporary loss of taste and smell. That is a very common thing. Like, I can say this, I can share with every one of you here right at this time, a point of time. As I told you, being a COVID survivor, I have faced each and every symptoms of these. This distaste and the smell, that is going away of the smell, it's a very, very, like, it's a traumatic symptom, I would say. This going away of smell is a very weird experience. Okay, so that is what, please check every time if you are coming across these symptoms. And that is what my suggestion to every one of you here. You are all responsible citizens. You're all responsible people of the world. So please take this as a serious alarm and get tested yourself. Okay, and definitely spread this awareness to your near and dear ones. Okay. The distribution of ACE2 receptors in different tissues may explain the sites of infection and patient symptoms also. Okay, so definitely, for example, if we talk about the ACE2 receptor, this ACE2 receptor is found on the epithelium. I hope everybody is aware of the term epithelial tissue. Epithelium or epithelial cells or epithelium of organs like intestine, or epithelial cells in kidney, in blood vessels, that explain the gastrointestinal symptoms and cardiovascular complications. So lymphocytic symptoms or endothylitis, lymphocytic endothylitis, the name of that, what to say, disease, we can say. It has been observed in even post-mortem pathology examination of the lung, heart, kidney, and liver, as well as liver cell who have died with COVID-19. Yes. So this is definitely a very important thing. I'll just write down here. It's lymphocytic endothylitis. So this has been detected in these organs which I mentioned right now, in lungs, in heart, in kidney, in liver, definitely in the postmortem reports of the COVID patients who died in COVID. Okay, so definitely these findings indicate that this virus directly affects these organs. Okay, and yes, the same was found with SARS-CoV-1 also, right? So their modus operandi remains the same, just their spread efficiency, their infectiousness, the capacity of infectiousness is definitely higher in this case. Okay. Now, next we have immune response and disease spectrum. So after the viral entry, the initial inflammatory response, it attracts virus-specific T cells to the site of infection, where the infected cells are eliminated before the virus spreads, leading to recovery in most people. Like in a survey in about 24 patients who develop severe disease, they elicit an aberrant host immune response. Definitely. You'll come to know across about cytokines. So definitely these cytokines are a direct involvement in this case. So we know that cytokines, they lead to inflammation. It's a major part of immune system. So we get to see this involvement of cytokines even in this immune response by COVID-19. Okay. And yes, like for example here, a very interesting example, post-mortem histology of lung tissues, as I just mentioned about that also, who died of COVID-19, they have confirmed the inflammatory nature of the injury with features of bilateral diffuse alveolar damage, hyaline, okay, membrane formation, interstitial mononuclear inflammatory infiltrates, and this desquamation consistent with acute respiratory distress syndrome. Yes. And all these were severe in the Middle East respiratory syndrome, that is MARS, that took place in Saudi Arabia around 2012. And basically a distinctive feature of COVID-19 is always a presence of mucus plugs in fibrinous exudate in the respiratory tract. And this mucus plug, no, this explains the severity of COVID-19 even in younger adults. You will get to see most of the younger ones, those who have died out of COVID-19, they mostly died of this respiratory trouble only. And it's due to this mucus plug formation in their respiratory tract. That's the history which is saying about COVID-19. And definitely this is potentially caused by the over production of pro-inflammatory cytokines, as I just now told you, that accumulate in the lungs, eventually damaging the lung parenchyma. Yes. So major infection or the major damage is caused in this particular portion of the lungs, and that is lung parenchyma. Why? Due to the overproduction of pro-inflammatory cytokines, because we don't need cytokines in much amount. We need cytokines definitely, but not in major amounts. So what happens in case of COVID-19 patients? There is major overproduction of the cytokines, and that is why these lung parenchyma, basically, this is the major area of infectiousness. The effect is caused in this particular zone or area. Okay? So just before going to the symptom cycle of SARS-CoV-2, I would also like to tell you that, yes, some patients also they experience septic shock. Septic shock. It's very interesting. So, and multi-organ dysfunction also. So, for example, the cardiovascular system, right, it's often involved early in COVID-19 disease. And it's reflected in the release of highly sensitive troponin and natriuretic peptides. Okay, so they are very, very important. And troponin is not at all good for cardiac patients. Yes. So see, there are a lot of phases in this case. It's not only about respiratory tract. That is why this disease is really becoming an alarming for us. It has multiple aspects of infectiousness. That is why everyone is now every sect of researchers, like they are now coming up with different drugs. Okay, so a lot of researches is going on. That is what the problem is with the SARS-CoV-2, as it's not restricted to only one site of organ infection. It has got multiple sites of organ infection. Okay, and cytokines, they mediate and regulate immunity, inflammation, hematopoiesis. Okay. And we know that cytokines in other organs in some patients also, they can cause extensive tissue damage. Okay, like thrombus formation. Okay, one of them is thrombus formation, which is very important. Like it can be organ dysfunction. So this also might happens. All right. So these things are very, very important. And yes, we have to take care of these things. So now, yes, this is the symptom cycle of SARS-CoV-2. As you can see, we just come across this terminology, SARS-CoV-2 viral load. So there are different phases of the symptom cycle, everyone. It's pre-symptomatic phase. Next, we have mild, moderate illness. Okay, so you can see the days it's going by and passing by. So with the increasing number of days, the illness is getting critical. So just when the virus has entered your body, when you even don't know, okay, you are among yourselves only, and you never know when and that virus has entered inside your body. So that is pre-symptomatic phase. Yes. Next, yes, the viral genome, it's there. It's the release of viral genome, release of viral nucleic acid. So this phase starts from the second day and it almost continues till the last day. So from 2 to 4, 8 days, it's mild, moderate illness. Post 6 days and 8 days, it's the severe illness band or zone. And next, it's critical illness. So can you see here, there is a overview. What should we do in these cases? So when it's mild, moderate illness, you can stay back at home and quarantine yourselves. When it's severe illness, it's time to go to hospital. Yes, hospitalization is mandatory at that point of time. And finally, when it's critical illness, we need to go for, yes, antibody check and ICU admission. That's the last phase we can do that. Okay, that's the antibody response to SARS-CoV-2. Okay, all right. I hope this cycle is clear to every one of you. So next is risk factors that are associated with the development of severe disease. And yes, these are very important things to be understood. That is older age, hypertension, cardiovascular disease, diabetes, obesity, and malignancy. They are definitely associated with these risk factors because clinical outcomes are influenced by host factors like these: older age, male sex, underlying medical conditions, okay, as well as the factors related to the virus, definitely virus in the sense the viral kinetics, host immune response, all these factors. Okay, so it's very, very important to know about them. And most importantly, there are certain laboratory markers that we use for COVID testing. So some of them are like raised C-reactive protein. We have C-reactive protein. Okay, another one we use commonly is ferritin. Next, we have raised IL, interleukins, basically interleukin 6. Okay, we have again raised lactate and lactate dehydrogenase test. Okay, these are the markers, lactate dehydrogenase. So all these are the different markers that we use: raised C-reactive protein, raised ferritin, raised IL-6, raised LDH. Okay, so yes, these are some of the cases as we can see, correct? That is, we can see this thing in front of us, how alarming. So we can see that Maharashtra is definitely the highest cases we have detected so far in Maharashtra. And even now in the second wave also in India, we are witnessing in this particular state. Okay. This is the overview of India I'm talking about. And yes, in India, the highest alarming state we have with Maharashtra. It's a state of India. Okay. You can see the reported cases. Okay. So yes, coming up to therapeutics currently under investigation, it's very important, right? So we are really confused about the drug aspects, which drugs to be administered. That is very important thing. So yes, entry to the cell, and when we talk about that is ACE receptor inhibitors. So viral replication will be RNA polymerase inhibitors, and such will be immunomodulators you have to use. So what is this basically? That how we can stop the entry to the cell. Okay, that is what is entry to the cell. So we have to think about certain ACE receptor inhibitors, right? Because ACE is the receptor for the virus. So we have to think about certain inhibitors that will be inhibiting the ACE receptor. And most importantly, we have the viral replication. So for the inhibitors that we generally be having as RNA polymerase inhibitors will be remdesivir, one of the most administered and registered drug that has been approved by WHO currently. Okay, and the host immune response will be with this, tocilizumab. Okay, next we have fusion inhibitors, we have ribavirin. Okay, these are all the drug compositions. In layman terms, they are the drug composition. Okay, so definitely they will be marketed in some different names. Okay, next we have umanofovir, and the viral replication inhibitor will be favipiravir. So these names and drugs are very important to be known. They are part of therapeutics that will definitely help in checking this spread and checking the disease, definitely. Okay, so this definitely is very, very important information for all of us. All right. So immune response to the virus and its role in protection. So we all know that COVID-19 leads to an antibody response to a range of viral proteins. But the spike protein and nucleocapsid, in the beginning, we come across these two proteins, right? They are those often used in serological diagnosis. And few antibodies are definitely there that are detectable in the first 4 days of illness. So basically, everyone, a wide range of virus neutralizing antibodies have been reported, and they are emerging evidence, we can say, that really suggests that definitely we can correlate with the severity. Okay, and the duration and the protectivity of antibody and T cell responses, okay, to this coronaviruses, they appear to manifest the cross reactivity with SARS-CoV-2. Okay, so that is what. So still we are in progress, definitely. That is what. Definitely for the serological diagnosis, like when we go for our antibody tests, okay, I think everybody is aware of this antibody tests. Antibody means, suppose you are an asymptomatic patient, and you later on even you realize that you might have this COVID virus in yourself. So if you do a antibody test, you get to know that yes, that antibody is there in your body, and basically at that point of time, which two proteins are uptaken? It's the spike protein and nucleocapsid protein presence. Okay, with this two, we definitely can realize, or we can definitely understand the presence of this virus in our body. All right. So that is what I told, that the duration and antibody T cell responses remain to be defined through studies with longer follow-up. So CD4 T cell responses to endemic human coronavirus, it meant its manifest cross-reactivity with SARS-CoV-2. But yes, their exact role, the CD4 T cell role in protection, it remains still unclear till now. A lot of researches are going on over CD4, the T cells, okay? So let's see what we come up with and what we end up with. All right, so it's almost, I have come to the end of the session. So I would like to end up my session with some future queries. And the future queries is just like that. What should be the window period of infectious patients? Because it varies from one patient to another. How can, or what is the protective role of T cell immunity in COVID-19? Or what are the determinants of healthy versus dysfunctional response in case of COVID-19, right? So definitely, yes, some questions will be unanswered till the end. And that is what, the understanding the pathogenesis of SARS-CoV-2 will be vital in developing therapeutics in future also. Yes, we definitely have come up with the two vaccinations, okay, but still a lot more to come up with. It's not the end here. We have to come up with some better therapeutics worldwide so that this can be curbed sooner. And more data are definitely needed to understand the determinants of healthy and dysfunctional response, even the immune markers for protection and the severity of the disease. And is, of course, neutralizing the antibodies are potential correlates of protection. But the other protective antibody mechanisms definitely can exist. Okay. And similarly, the protective role of T cell immunity and duration of both these antibodies and T cell responses, it can be defined, right? So that is what. Greater the understanding is needed regarding the long-term consequences following acute illness, and this multi-system inflammatory disease will be definitely be answered. Correct? So yes, with this, I would like to end up my session on a positive note once again. So this is the COVID vaccine. This is the statistics I have shown up for India and global aspect. So we can see this is the total number of population, those who got till now the COVID vaccines. Okay, so it's definitely a huge, and we are trying, all of us are trying our level best to achieve the best in future too, right? And this result in this stats is till 2nd April, it's updated till 2nd April, and that is what I have shared with every one of you. So this is what. And yes, to conclude our session, that is what recent evidence, it ushers that definitely appropriate immune response is fundamental to COVID-19 pathogenesis. But a lot of things remains unexplored, right? So we have to do that. Everybody has to do that. And with this positive mindset, we can say that, yes, one day will come, then we'll definitely enjoy, then definitely we'll all will get this fear out of themselves that yes, no more COVID tension resides among us. Thank you so much, folks. Till this, I would like to uh, end up my session. I hope everybody liked it. It was beneficial, definitely, to every one of you here. Thank you so much.
It's amazing, ma'am. Thanks for giving your valuable insight on SARS-CoV-1 and SARS-CoV-2. We are living in a unique time right now, and I also felt so disturbed when government announced lockdown and about
Yes,
public gathering place. But your valuable insight will give us and our audience a clear concept why government announced this type of lockdown. So
definitely,
we have
definitely.
to be more conscious about transmission of corona and COVID-19 also. That's similar.
Definitely.
Sorry to hear that, ma'am. You are also a survivor, and
Yes,
congratulations you because you are front of us and giving
exactly.
us a
That is why I mentioned that yes, I am a COVID survivor and nothing to worry about that. Just you need to keep safe, and that is what, and just that is what you have to quarantine yourself. Okay,
yes,
just
ma'am.
get tested, quarantine yourself. That is the ultimate thing you have to do.
And ma'am, congratulations also you,
Thank you. Thank
because
you
you
so
are
much.
front of us and you're giving us a valuable and important information about COVID-19. Thank you, ma'am.
Thank you so much,
Um,
Salma. Thank you. It's my pleasure and honor too.
it's time to move our next speaker. Our next speaker is Arvana Chatterjee. So let's start.
A very good evening to everyone. Myself, Arnaba Chatterjee. I have done my BSc in Microbiology Honours from University of North Bengal, and I have pursued my Masters in Biotechnology from Jadavpur University, and currently I am working as a Senior Assistant Teacher in West Bengal Government Secondary School. I am also a very avid reader, and I like to go through all the recent researches related to cell biology and molecular biology. So let's see you in the webinar. Thank you so much. Hi, good evening.
Hello,
Hello.
ma'am.
Yeah.
Hello, ma'am.
Yeah, very good evening. Nice to see you. And I should say that it was a fantastic speech and it was very informative speech by Priyanjana Di. She is very close to me because I know her personally. We have studied Bachelor of Education together. And since then, we have been good pals, I would say. So it was a fantastic speech by Priyanjana Di, and now I would like to start my part of this webinar.
Yes,
So
ma'am. Now still is yours.
yeah, thank you so much, Salma. So I would say a very good evening to the audience watching this webinar, and I would like to extend my heartiest thanks to the organizers of this event at Institute of Global Professionals for giving me this opportunity to speak in front of this awesome audience. Myself, Arnaba Chatterjee. Basically, I am a life science teacher. As well, I am a curious student of life processes and the enigmatic world of science. I'm really in love with science because I think from my very childhood, I have been fascinated by scientific processes. Let me now take all of you to a tour of my topic of discussion for this evening. So we know that the year 2020 witnessed an overwhelming situation where the whole world became grasped in the clutches.
Hello,
Hello?
ma'am.
Yeah,
Need to
yeah.
share your slide, please.
Oh, okay. I just forgot. Let me share my slide. Yeah, yeah. Yeah. Okay. Yes, yes. I just forgot. Sorry. Apologies, actually. Yeah. So I would share my slide, entire screen. Okay. Share. Okay. So It's opening. Just give a second.
Yeah, no problem.
Yeah. Can you see, sir? Is it
Yeah,
visible?
yeah, I can see. Yeah, just press F5.
You can. Okay. Okay. Press F5. Okay. Fine. Okay. F5. Yeah.
Yeah,
Okay.
okay.
Okay. Fine. Hmm. Okay. So, so can I continue? Hello, sir.
Yes.
Hello.
Yeah, you can continue.
Oh, okay. Okay. Fine. So, as I was saying, the year 2020 witnessed an overwhelming situation where the whole world became grasped in the clutches of a deadly disease, COVID 19, which is essentially severe acute respiratory syndrome and is caused by the novel coronavirus 2. The disease got such a name because it was first detected in the Wuhan province of China in December 2019. Now, as the name suggests, COVID 19 causes severe respiratory distress and even death in some cases if left untreated. The incubation period, that means the time from when the virus enters the host, like the person, susceptible person, and the onset of infection, this is called the incubation period. The incubation period of this disease is normally 14 days, like 2 weeks, but the symptoms peak at about a fifth day from the onset of the disease. Like you get to see the symptoms, like in most cases, people show symptoms, but in other cases, there are people who are actually asymptomatic, and then it's a very problematic situation because you never know that you are harboring the virus yourself and you are spreading the virus because you're talking with everyone, you are moving in the market, you're going here and there, and you're spreading the virus. You're sharing it, and rest others are getting affected by the virus. So, in this way, COVID 19 has so far claimed millions of lives worldwide, and it is still a threat to the people. Therefore, the situation warrants effective vaccination strategies to be employed in order to ameliorate this deadly disease. Now, speaking of vaccine, I feel it is very much important to know in detail about a vaccine, its types and mechanism of action. So, let me now start my presentation. So, first, we have to get a brief idea about the history of vaccination and how actually vaccine developed. So, vaccine actually developed due to the groundbreaking work of one very good person. I should say he was a doctor actually, Dr. Edward Jenner. He resided in England. He in 1796 he was working with smallpox disease, which was a very bad disease. Now it has been eradicated from the whole world, but then it claimed millions of lives at that time in 1796. At least 400 years earlier, I'm talking about. While he was working with smallpox disease and the process of variolation. Now, what is variolation? Variolation is actually injecting the virus or infectious fluid into a healthy individual to foster the development of the disease symptoms and hence get the person vaccinated against that disease. So the word vaccine originates from the Greek word vacca. Vacca means cow.
I'm
In
sorry
order
to
to,
disturb again.
hello,
Hello. Sorry to disturb again. Your voice is not clear. Right?
okay, okay. Can't you hear me now?
Now is it okay? Now is it okay?
Okay, okay. So you couldn't hear me till now,
No, no, I can hear. Your voice is
sir.
like some chubby. That's the reason. No problem. But you can continue right now.
Okay,
My voice is clear.
okay, okay. It's audible. Okay. So let me again restart. So I was talking about the history of vaccination and vaccine development. Apologies for my voice breaking. I didn't know that there was some technical glitch. Sorry. Vaccine developed due to the groundbreaking work of Dr. Edward Jenner around 1796 while he was working with smallpox disease and variolation. Now, variolation, what is the process of variolation? Variolation is actually the act of injecting infectious fluid to a healthy person to foster the development of the disease so that the person can get immunity against that disease. So the word vaccine originates actually from the Greek word vacca, meaning cow, in order to mark Jenner's work related to smallpox vaccine. Now, why this vaccine originates from the Greek word vacca, meaning cow, or what is related to cow, we would see in the next slide. So Edward Jenner and the advent of the science of vaccination actually deals with milkmaids. Actually, he was a physician. He was a doctor, and he was studying extensively about smallpox disease, and he found out in studies that milkmaids, the people who milk the cows, developed a rather mild related disease called cowpox. And upon further investigation, he concluded that the virus causing cowpox disease was very much related to the virus which causes smallpox. So they are antigenetically related. The cowpox virus and the smallpox virus, they are related. So in order to testify his findings, he inoculated. I said that he injected a small boy. His name was James Phipps with fluid from a cowpox pustule. That is the acne. That is the person who has developed the disease cowpox has some rashes, has some inflamed, raised pustules or nodules on his or her body, and from an infected pustule, he injected the fluid into a small boy, and as expected, the boy contracted the disease, and in this manner, he was vaccinated against smallpox because, as told earlier, cowpox and smallpox were related antigenetically. So next slide, actually. So, sorry. How to go to the previous slide? Sir, how to go to the previous slide? Hello. Uh, Actually, my goodness, this is. Let me do some back. Okay, okay. I got it myself. So, so here is a picture of Edward Jenner and the process of variolation being performed by him. He's injecting the virus to this boy. I say the boy's name is James Phipps, and he vaccinated the small boy. He was actually 12 years old then when he received the vaccine. So, and this was actually the Victorian era. So you can see the English people used to dress up like this. So very good information regarding vaccination, and the history is very fascinating. Okay, so in this way, the vaccine developed from this Greek word vacca, meaning cow, in order to show some tribute to this Jennerian vaccine development process. Okay, so now let us have an overview of this vaccine. So, what is a vaccine actually? A vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. So, I would discuss in detail all these terms: active acquired immunity, how immunity is achieved against a disease, what is immunity. In later slides, let us first discuss about this vaccine. So, actually, a vaccine, the composition is typically it is an agent that resembles a disease causing microorganism. It may be a disease causing microorganism itself, or it may resemble a disease causing microorganism and is often made from attenuated, that is modified, changed, or killed forms of the microbe. Microbe means small microorganisms, small organisms which we can't see with unaided eye. We have to use the microscope. It's toxins. If you know what are toxins, the toxins are actually the proteins released by um, microorganisms which cause disease or which cause discomfort in the susceptible individual, or one of its surface proteins. So proteins are actually most of the toxins are made up of proteins. I would say that all the toxins are made up of proteins. Then this agent, the vaccine agent, stimulates the body's immune system. Immune system is actually a battery of cells and molecules which actually provide a defense against foreign antigen and viruses and microorganisms, so that we don't get ill. Suppose if our body had no immune system, then each and every day we would have contracted disease, and we would have been sitting in the hospital each and every day. So thanks to the immune system of the body, that these agents are recognized as threat and they are being destroyed. And the immune system also has this memory to further recognize and destroy any of the microorganisms of such type, like associated with that agent that it may encounter in the future. So immune system is a very specialized mechanism of the body. It is actually having specificity. It has antigenicity, and it has reactivity, and also a memory, an immunologic memory that it recognizes that previous infection and protects the body from further infection. So for instance, if somebody is affected with like this measles, mumps in childhood, the person will not get affected in later stages. Why? Because of this immunologic memory. Because the immune system has remembered, on that day in the very childhood, this person got affected with this disease. So if the virus comes, then I will destroy, and I would not let the person get affected once again. So now let us move to vaccine once again. Depending on the role, vaccines can be prophylactic or therapeutic. So what is prophylactic? Prophylactic vaccines are designed to build immunity in a patient. A prophylactic is also known as a preventative vaccine. It involves introducing antigens into a person's body. The goal is that the individual's immune system will create antibodies for those antigens and become immune to the associated disease or illness. A therapeutic vaccine, on the other hand, is a vaccine which is administered after a disease has occurred or an infection has occurred. So it is actually different from the prophylactic vaccine. It works by activating the immune system of a patient to fight an infection. Now, let's move to the next slide. So, since I'm from India, I have the picture of India's newly launched COVID vaccines. We have Covaxin and Covishield. So these vaccines have passed through all the test and scrutiny processes in the laboratory and are now available to the mass for vaccination purposes. Next slide, I would say. So this is the COVID-19 vaccine in general. This is the photograph of a COVID-19 vaccine vial. The process of administration of vaccines is called vaccination. Vaccination is the most effective method of preventing infectious diseases. When a child is born, the child is administered DPT vaccine, that is diphtheria, pertussis, tetanus vaccine. Then up to 5 years of age, the child is given doses of polio vaccine. Why are these?
Hello, ma'am.
Hello,
Ma'am, your audio is not clear.
can't you hear me?
Now is it clear? But I don't know why is it facing some problem with the audio. Now
Can
you
you
can
hear me
continue.
now?
I can hear you.
Uh,
Now is it okay?
See, yeah, whenever my voice breaks, please inform me. Okay, sir. I don't know that,
Okay. Now
like, when it breaks.
no, I understand, understand. But
Yeah,
now
yeah.
is it okay? Is it okay? Far better.
Oh, okay, it is okay. Okay, it's better. Okay, fine. Thank you. Apologies once again. I don't know what is
No.
happening with this thing.
You can check with without
I
headphone
don't.
also. Let you check without headphone.
I have checked my headphone. It's new. It's a brand new headphone. Now, if it is not working in this case, then my I don't know what is happening. Oh, can you hear me now?
Okay. Yeah, I can hear you.
Uh, Okay, fine. So, folks, where was I? Okay. I was telling you about the child being vaccinated against many diseases like polio vaccine. We have diphtheria, pertussis, tetanus vaccine. Then we have MMR vaccine. That is the mumps, measles, rubella vaccine. So these vaccines are provided in the childhood to boost the immunity so that the child doesn't get infected with these common diseases. Now, the World Health Organization, WHO, which is actually abbreviated as WHO, reports that licensed vaccines are currently available for 25 different preventable infections. So, okay. Now, the goal of vaccination regime is actually to produce immunity, to foster immunity. Immunity means the presence in a person's body of previous cells and substances known as antibodies that can produce a protective immune response against antigens or microorganisms or foreign viruses. The immune cells and antibodies attack viruses and bacteria or the toxins, that is, the proteins they produce, stopping them from multiplying and causing disease. So, vaccination is very important and it should be very sophisticated. Now, what are the types of immunity? We have actually two types of immunity. Active immunity, active immunity and passive immunity. Active immunity is again divided into natural active immunity and artificial active immunity. A passive immunity is in that case also divided into natural passive immunity and artificial passive immunity. In the next slide, we have this active immunity. We have this classification: natural active immunity, which results from exposure to foreign microorganisms and subsequent infection, is the natural active immunity, which we people get naturally. Artificial active immunity is the immunity which results from vaccination. Like if we get the COVID vaccine, then we won't be affected by, or we expect not to get affected by coronavirus. So that is artificial active immunity. Next, we have passive immunity. Natural passive immunity is the immunity which results from passing of maternal antibodies to newborn through placenta. That is the organ where the newborn is actually developed in the mother's womb or breast milk. Like the secretory IgA antibodies are passed to the child when the mother feeds the newborn. So these are the methods of passing of natural passive immunity. And artificial passive immunity is actually a very new and sophisticated concept because it results from monoclonal antibodies, and monoclonal antibodies are actually a part of an area of research in biotechnology. And many vaccines have been developed with this monoclonal antibodies, MABs, which we call. Biotechnologists and geneticists are working with monoclonal antibodies to develop a lot of good vaccines. Next, we have to get to know about herd immunity. Herd means group. Herd immunity is actually very important for vaccination purposes because it is actually the immunity of the whole population. Herd immunity, as we know, as we can see in this particular slide, also called population immunity, is an indirect protection from an infectious disease that happens when a population is immune, that is protected either through vaccination or immunity developed through previous infection. So see, in this particular picture, we have all these people. So first we are having this the blue people are actually the susceptible people. Then we are having this yellow people who are the immunized people, and we are having some red people who are actually contagious. So how this thing passes? Infectious agent, in case of herd immunity, if herd immunity is not there, then infectious agent passes freely from contagious to susceptible, making that person, that susceptible person contagious again. But in case there is herd immunity, like most of the people of the population have been vaccinated against coronavirus vaccine, then the contagion cannot freely pass via immunized to susceptible. See, the arrows are being blocked. So the contagious person now is contacting an immunized person. Suppose I am telling you, I have been vaccinated. I have received the vaccine, Covishield vaccine. So if somebody comes near me, if the person is contagious, has the virus, I won't be getting the virus because I am already immunized, and I won't be passing the virus to susceptible people. So as we can see in this slide, when most of the people of a particular population gets vaccinated, there develops an immunity called herd immunity. So when there is no herd immunity, the virus can freely pass from any direction, straight, diagonal, whatever direction the virus feels it can pass through. But if the herd immunity is developed, like all the people who are having this who are being labeled in black, deep blue, they are actually having been vaccinated, and so herd immunity prevents this passing of disease from susceptible to from infectious to susceptible person. So this is the process of vaccination. Vaccine is given generally by injection. I am actually a very I feared injection, but when I got vaccinated, I didn't feel anything. I was asking the nurse, like, have I been vaccinated? The injection, I couldn't feel the pain actually. So it was a very good experience in my case. And the vaccines contain weakened or dead microbes to elicit a normal immune response. So let us have a look into the types of vaccines now. We have inactivated vaccines which contain inactivated but previously virulent microorganisms that have been destroyed with chemicals, heat, or radiation. Example, IPV polio vaccine, that is the injectable polio vaccine. Attenuated, the term attenuation means modification. Attenuated means some vaccines contain live attenuated microorganisms. Many of these vaccines are active viruses that have been cultivated under conditions that disable their virulent properties. Means they can't affect, can't cause their pathogenicity is destroyed. They can't cause disease if injected to an individual, or that use closely related but less dangerous organisms to produce a broad immune response in an individual. Example, the tuberculosis vaccine, that is a Bacillus calmette variant, there is a BCG vaccine available. Then types of vaccines continued. We have 8 or 9 types of vaccines. We have another vaccine called viral vector vaccine, which uses a safe virus to insert pathogen genes in the body to produce specific antigens such as surface proteins, markers to stimulate an immune response. Then we have toxoid. Toxoid is actually very popular vaccines available. Like we have diphtheria and tetanus vaccines, which are toxoid vaccines. Toxoid vaccines are made from inactivated toxic compounds that cause illness rather than the microorganisms. Example, tetanus and diphtheria vaccines. If you remember, guys, I was previously talking about therapeutic vaccines. Like I was talking about the vaccine which is administered after the disease or infection has occurred. So when we get a cut by a rusty nail or something, we say that tetanus might occur. So in order to prevent that situation, we get the tetanus toxoid vaccine. vaccine, that is the TET-vac, which is actually the tetanus toxoid vaccine. Next, we have a sophisticated type of vaccine, which is actually a very modern vaccine called surveillance vaccine. After the advent of biotechnology and genetics, these type of vaccines have been made. Rather than introducing an inactivated or attenuated microorganism to an immune system, a subunit vaccine uses a fragment of it to elicit an immune response, to foster an immune response. Example, human papillomavirus vaccine, which causes cervical cancer. Okay, cancer of the cervix. Okay, next we have the conjugate vaccine. Now, conjugate, what is the meaning of conjugation? Conjugation means addition of something to facilitate the response. So, certain bacteria have a polysaccharide outer coat that is poorly immunogenic. That is, in itself, it can't elicit an immune response. But by linking these outer coats to proteins, the immune system can be led to recognize the polysaccharide as if it were a protein antigen. Example, Haemophilus influenzae type B vaccine. Haemophilus influenza type B vaccine. Next, we have heterotype B vaccine. That is a heterologous vaccine, also known as Generian vaccines. Remember Edward Jenner, the person who developed vaccine? Are vaccines that are pathogens of other animals that do not cause disease in humans. Example, Bacillus calmette guerin, that is a BCG vaccine used in tuberculosis. RNA vaccine, so the our hero of today's webinar, COVID-19 vaccine, is actually an RNA vaccine. It is a messenger RNA vaccine. It is a novel type of vaccine which is composed of the nucleic acid RNA packaged within a vector such as lipid nanoparticles. So the COVID-19 vaccine is actually an RNA vaccine. We have DNA vaccines. We have other vaccines, sRNA vaccines, but we would not go into such details because those are not important for this webinar. We have to deal with this RNA vaccine, that is the COVID-19 vaccine. Let's check out the next slide. So the COVID-19 vaccine development process is fully automated and sophisticated machines are used to produce high efficiency vaccines. PCR is done, that is polymerase chain reaction is done, autoradiography, crystallography, etc., electrophoresis, everything is done. Then the process of vaccine development also has this gene sequencing, packaging into viral vector, and then insertion. All these are done. Then also adding such surfactants or surfactant proteins or those things which actually have buffer solutions to provide stability to the vaccine. So, so this is a picture of employees preparing themselves before getting inside a lab where Covishield, prepared by AstraZeneca, Oxford. COVID-19, this is the virus vaccine, is being manufactured as India's lab, that is the Serum Institute of India, Private Limited. So in this process, like the people, the researchers have, the researcher is actually using the sanitizer and he is wearing the personal protective equipment, that is a PPE, having mask, having shield, everything is there. So so safety level is also monitored of these vaccines, and also the process is highly like people are also taking precautionary measures to not get infected by the virus themselves. Okay, so let us have a detailed look into the two vaccines developed by India, the Covishield vaccine and the Covaxin. The Covishield vaccine, I have been vaccinated by this Covishield vaccine. So the qualitative and quantitative composition of this vaccine is actually one dose, that is 0.5 ml, contains Chidox 1 NCOV, that is the COVID-19 coronavirus vaccine, recombinant 5 into 10 viral particles. So there are 10 viral particles. So here it is to be noted that recombinant replication deficient chimpanzee adenovirus vector, not chimpanzee DNA, but chimpanzee adenovirus vector encoding the SARS-CoV-2 spike S class glycoprotein, which is actually the immunogenic, or which is actually the pathogenic molecule of the coronavirus because coronavirus got its name due to its crown-like structure due to the spikes produced in genetically modified human embryonic kidney cells, that is HEK293 cells. So this is the picture of this Chodox N-COVID-19 coronavirus vaccine recombinant Covishield. Covishield is manufactured by Serum Institute of India Private Limited, available in 5 ml or 5 ml 10 doses vials. Okay. Then we have the co-vaccine. Again, co-vaccine is also similar. It contains 6 micrograms of whole virion infected. Here, actually, it is not vision; it's virion. I'm now getting into the thing, where an inactivated SARS-CoV-2 antigen. The strain is actually NIV 2020-770, and the other inactive ingredients such as aluminium hydroxide gel, 250 micrograms TLR, that is toll-like receptor 7-8 agonist. Imidazoquinone, 15 micrograms, and 2-phenoxyethanol, 2.5 micrograms, and in phosphate buffersaline, 0.5 ml. Covaxin has a beautiful look. So it is manufactured by Bharat Biotech Private Limited in India. So let us have an exclusive tour of the COVID-19 vaccines available all over the world. So vaccine picture gallery, we will be looking at all these vaccines which are being developed. So, so the first vaccine is Pfizer COVID-19 coronavirus vaccine. It is available in the United States of America. Next, we have this Pfizer COVID-19 vaccine. Next, we have AstraZeneca. It is now available all over the world. Next, we are having Johnson & Johnson COVID-19 vaccine. It is available in South Africa. Next, Russia has the Sputnik 5 COVID-19 vaccine. It's available to the Russian people. China, the main hero, the main protagonist, where the COVID-19 developed, the coronavirus originated, has developed 2 vaccines: CNPG COVID-19 vaccine and another vaccine named CanSino Bio. So these 2 vaccines. Next, we have Moderna company. Moderna Research Institutes have developed COVID-19 vaccine for the UK population. Pakistan has received its Sinopharm vaccine. Bangladesh has received COVID-19 vaccine from India, namely Covishield, because Bangladesh is a friend of India. Now, we have to know who should get COVID-19 vaccine. There are some certain theories of each and every people. As general folks, as general people, we need to know who should get COVID-19 vaccine, who should not get COVID-19 vaccines, and other facts. So COVID-19 vaccine can be safely administered in all individuals 18 years old and beyond. Each COVID vaccine developed is thoroughly tested on healthy volunteers, healthy people, and have passed all stages of scrutiny and scrutiny in drug development. So it is completely safe to get vaccinated by any of these WHO, that is World Health Organization recommended vaccines. So first in the vaccine development process, the vaccine is developed. Then there are some phases. The first trial is in guinea pigs or like rats, like mouse. Then another trial in second trial in those susceptible animals, and next trial in humans if there are some volunteers. And then after another scrutiny, another check, the vaccine is actually released by the World Health Organization, give the license to release these vaccines to the companies creating the vaccines. Okay, and then who should not get COVID-19 vaccine? Well, one should not get COVID-19 vaccine if one had a severe allergic reaction to any of the components of the vaccine. So the person, so actually normal people do not know, but the people who are aware will check the components, the ingredients given in the vaccine vial or the manual which one can get with the vaccine file. So there, all the ingredients are being listed. Go through the ingredients. If you know that you are having any allergy to any of the ingredients, you should avoid that vaccine. Or you had a severe allergic reaction after the previous dose of the vaccine. So actually, there are two doses of vaccine. The first dose, after the first dose, you developed hives, chills, fevers, allergic reactions, raised rashes. You should avoid that vaccine. Or you are currently ill, you are having an acute infection or fever, you should not get the vaccine. What should you mention to your vaccination provider before getting vaccinated? When you are getting vaccinated on that day, when you are visiting the hospital, then you should be informing your healthcare provider or the nurse who is administering the vaccine to you that whether you are on a regular medication for any illness or for how long and for which condition, as in it is not advisable to get vaccine in such ailments. So if you are having any disease, a debilitating disease, you should inform. Or if you have any sorts of allergies as told previously, or have fever, or have a bleeding disorder like hemophilia or on a blood thinner, or are immunocompromised, have any organ transplant operation done on you, or are you pregnant or lactating, or have received another COVID-19 vaccine, not this vaccine. If you are having getting vaccinated, then you should get the two vaccine doses from the same vaccine. Like I have got the two doses from Covishield, not that I have got the first dose from Covishield and the second dose from Covaxin, because that would mess up my this thing. Okay. So route of administration of COVID-19 vaccine and generally all vaccines is intradermal route by injection to the deltoid muscle of the upper arm. See, you can see in this picture, the girl is very happy. I was not happy when I was getting vaccinated because I was apprehending that I might get a very sharp feeling, but nothing happened as such. Okay, so dosage of administration of COVID-19 vaccine. The first dose on any day by visiting the nearest healthcare setting or hospital. If you are healthy and hearty, you can go on any day. And second dose normally after 4 to 8 weeks, depending on the brand of vaccines administered. Now, benefits of COVID-19 vaccine, there are a lot of benefits, but people are actually not aware. So, vaccine reduces your risk of infection. Uh, I would not say that vaccines can actually nullify the chance of infection, but it actually reduces your risk of infection because there are some cases where after getting vaccinated, people have developed disease. But in that case, I would say that vaccine was not good, or rather, the vaccine was not injected properly, administered properly. The vaccine can help your unborn baby or newborn by creating this passive immunity. Okay, that is the natural passive immunity. I told about placenta and mother-child connection, breastfeeding and mother-child connection. The vaccine protects against severe illness. The vaccine will eventually help us ditch the mask because all over the world we are moving with masks, and so eventually a day will come. Let's be positive. Let's hope for the best that the vaccine will completely eradicate this disease as smallpox has been eradicated. The vaccine will also help you reconnect with friends and family. You need not get quarantined further. You need not isolate yourself. Spend good quality time with your family, with your friends. friends, family, and colleagues. Next, I'm actually on the end of my speech. If there are some side effects of COVID-19 vaccine, there are side effects of all drugs being developed. So there are also side effects of COVID-19 vaccines, but they are not actually adverse. They're very mild. Like you can get some injection site pain. Like for my example, I couldn't realize the injection being given, but after that I had some pain in my upper arm. There was then there could be a swelling, there could be some redness or erythema, there could be some itching also. You could get a headache, you could have a fever, you could have a body ache or malaise, you could feel like vomiting or nausea, you could vomit and you could have rashes. But these are actually very mild and very negligible. So now let's have this look into the future prospects of COVID-19 vaccine. As the vaccine has been developed, so we know that COVID-19 disease has claimed a lot of lives. But the aftermath of this pandemic shows a silver lining, a ray of hope, as most of the countries have successfully developed strong vaccination regime, and the doctors and healthcare workers have toiled a lot to cater to the basic needs and health of common people. The future beckons with the hope of heightened immunity power and disease control by the administrators and all concerned. We have seen that people have get vaccinated. There are a lot of people who are getting vaccinated each day, and we hope the cloud of discomfort will soon go away, bringing along a shine and bright morning of health and hygiene. Thank you very much for listening to my speech. Thank you very very much. So I end my speech with this thank you note.
Thank you, ma'am. That's so informative. Some of us vaccinated but
Thank
didn't know
you.
about the some of us vaccinated but didn't know about the history of the vaccine uses, types, why vaccine is important, and also about the vaccine fear of injection. So I think
Yeah,
this session
I
and
have this fear of infection. I have this fear of injection, but I didn't realize the injection being given. That was a good thing.
ma'am, I learned a lot from you, from your session, and I think audience also made their learning goal. Thank you, ma'am.
Okay, Salma.
Ma'am, now it is time to answer the question of audience. The question are shown in the screen.
How to save the babies.
How
Yeah,
to
see,
save the babies?
yeah, continue
Yeah.
on.
No, you continue, Udi. You say, then I'll say.
See, I'll be sharing a very honest and experience of mine. I have a 2-year-old kid of mine, and when we got this COVID positive, no, including him, he was also tested positive. Okay, so the thing is, yes, babies they don't undergo the test, but it's quite obvious if a mother is having if she is tested positive, then the doc says that automatically your child will be positive. So they are fast recoverer. Okay, they actually recover very speedily because their immunity power is better than us, believe me. And yes, there is no as such way of uh, like, even it's being told that below the age of 2 years you cannot even put the mask on their face. That is also not permissible. Okay, so just don't worry, they'll be having the common symptoms, the fever, the common cold, they will be having that. And after a span of 3-4 days, they will be absolutely fine. So actually, we should not be worrying about the babies. Rather, we should be worrying about ourselves. Okay, that is the age 45 and above is definitely very crucial and a very sensitive age group. Thank you. Honorable, you can continue.
Okay, so I agree. I completely agree with Priyanjana Di. So actually, as the government has advised that first the old age people like above 60 will take the vaccine, then the 45 uh, age group people will take. So actually, babies have a greater immunity power than us. So babies are less susceptible, and if they get, they will recover because they have a greater immunity power. So not to worry about babies.
Okay.
Okay.
The next question says, what will happen if you got vaccinated with two different COVID-19 vaccines? See, okay, then I guess I am reading I'm pronouncing your name correctly. It's Alizia. Hopefully I pronounced your name correctly. If not, please pardon my mistake. The thing is, it will not be happening with you, right? So whenever you are registering, okay, yourself with any COVID center who are like administering this particular vaccine, they won't be providing you with that. Like, supposedly if you are getting registered with any one of the center, they will be providing you with any one of the vaccine types. Okay, and it's a registered thing. Like in any country, whenever you are getting this particular dose of shots of vaccine, your name is already registered over there. So one person cannot get it is actually 4 shots if you are getting 2 doses of the same vaccine. So again, if you're taking up another vaccine, it will be equivalent to 4 shots. So that is not at all permissible also. So definitely your name will be registered. So in no way you will be getting 2 doses of 2 vaccines. Okay. Yes or no, if you have to say anything, you can please.
Yeah, I again agree with Didi. Actually, when you are going to the vaccination center or hospital, then they will be giving you only one type of vaccine. So, and they will be marking your name. So, if you go and if you feel like I want another vaccine, then I'll be getting super immunized. That will not happen. Rather, it's actually not. It's not recommended, and you should not get. And also, there will be near to impossible chance of you getting vaccinated with two different COVID-19 vaccines. That is next to impossible. So please don't worry, Eliza. Uh,
Yes, I guess there's another question. Do you think the vaccine is the only solution to solve the increasing cases of COVID? No, not at all. Because it's not like that. Yes, vaccination is the first shield of prevention towards this pandemic. And the point is that, no, we are actually standing in a very crucial timeline, and we are not definitely sure about that. Yes, we can believe in what is happening. Okay, so we have to trust this system, what is going on. And yes, definitely we can expect that, yes, vaccine can resist or it can control the increased case of COVID. But apart from that, it's not only about vaccination. We have certain drugs that has also been approved by WHO, okay, World Health Organization. So whoever is getting positive or getting tested positive, even with the proper dose of those drugs also, we can even control this particular COVID disease also, and yet to come in the future. Okay, so yes, whatever it's there with us, with that definitely we can at least control, even not 100% we cannot control that, at least 50% we can control that. Yes. Yes, continue on about you.
Okay, so the thing, yeah, vaccination cannot provide you with everything. You have to get social awareness, like you have to follow social distancing, wear mask, use sanitizer, and moving to the next question, which vaccine is most effective? Any vaccine recommended by WHO, that is World Health Organization, is effective. Not that which one is most effective. All vaccines are as good as. Over to Priyanjana.
Yes. No, no. I guess you have answered his question.
Yeah, yeah.
Yes. Any more questions? Yes. Why there are some people? Uh, See, I
Yeah.
want to answer this. Like, why there are some people who got vaccinated, but see, we have to judge all the parameters in this case. It's not that, like, even no, in our when we are kids, no, we had our vaccines allotted for us, like BCG vaccine. I guess everybody had this shots of all these vaccinations, BCG or even chicken pox. So can we assure that if you are taking up such vaccination, you will not be getting this disease in future, or you will not die? It's not like that. These modes, they are therapeutics. Vaccines, drugs, they are all modes of therapeutics. They lessen the chances of occurring these diseases, but we cannot definitely assure that once I get the vaccine, it's pretty sure that no, I cannot die of COVID-19. It's not like that. Even if you have no, like high density or high form of morbidities, so definitely it can take away your life too. So it's not like that. So we have to think positive. Yes, we should be getting these shots, so that yes, we have this lesser chances of this dreadful virus in the future. Okay. I hope I have answered. And yes or no, if you have to add anything, definitely you can.
No, I don't think that nothing has to be added to this thing which you have said, but you cannot guarantee that you will not die of COVID-19 as said. But yeah, there are certain physical conditions of people, like they are their body has not responded to that vaccine. In that case, they can die or they can get ill. But on a positive note, the higher are the chances that the disease will not occur, and the vaccinated people will be safe.
I guess Rowena Ornoba ma'am has already answered your question previously. Like she already discussed about this aspect about, we cannot say this is the best vaccine, because till now we are, this entire world is on a chance. Okay, everybody's taking a chance. Okay, we cannot assure that yes, this vaccine is the best. Even it's, the thing is, the disease itself has it's the tenure of this disease is still 1 year only, and vaccine has come up, it's for only few months, so we cannot recommend on that truly. Okay, so we have to believe, that is what we can do. Every vaccine, we have to, whatever vaccine is coming up for the respective countries, we have to believe on that, and we have to get that in. That is, we can do that, the best we can do.
Yes.
Today we learned a lot from both of you speakers. Our
Thank you.
pandemic situation is
Thank
the
you so much.
off situation because we lost our lover one, and we also survived many things in this situation. So, but we
Right.
have to believe. I think you
Right.
all are benefited by this session's audience. And dear knowledge seeker, I would love to thank you all who always join with us and also remain in till the end. Hopefully, you learn something new and you will implement it in your life. Please leave a review or you comment on our page. And if you learn something or we will help you a little bit, please spread the information with your friends. We hope to see you all again next webinar. Thank you so much. Stay happy, stay safe.
Thank you
Thank you.
so much.
Thank you, every one of you.
Because actually, I am the one who interrupt the session at the end of the session. Uh, Actually, thank you so much. But I have to consult personally with you guys, because I heard something interesting about Corona. So I'm so much confused about that, because I'm so much not confused. I'm so much worried about that.
It's not about not worrying anything. Like you have to be just follow and the basic guidelines. That will only, I guess, it will work.
Sure, sure, definitely it will work. But after that,
Yes.
as a human being, I have to worried about my total condition. I'm thinking about my immunity power. Is that still available or
Yes.
not?
Yes. That is what you have to take in good food, right? Proper food. You have to, what to say, proper physical exercise just to increase your immunity. That is what we can do. Yes, now we have these vaccines, but still, no, we are now aware of the second wave of this corona. And that is what
But
we never know what
one
will
thing is
happen.
that in India is a good condition right now. They're in the
It's
number
not
of
at all good in India.
no, actually it's a good in India, 'cause, by the information of the some state, in India the number of cases is so much increasing, but the death ratio is zero.
Yeah, the death rate has it's minimized a lot because no, that is what Arnoba has discussed also in our discussion about herd immunity. So it's actually working, we would say. So initially, when it started, when all the story of COVID-19 started, we had a huge death rate. Not huge, I would say, because it's not like Ebola. We had seen the death scenes of Ebola. It was not like that, but still yes, there were death cases, but now it has reduced a lot. But that is what I mentioned in my lecture also. The spread efficiency of this virus is it's too much. Okay, that is what it's
Yes, yes.
happening. Yes, that is what it's happening. So that is what, if we just control ourselves, wearing mask, becoming like nowadays we see that people have they have already dropped that idea of wearing the mask whenever they are going out publicly. So we shouldn't be doing that. Simple things we have to follow, right? So I guess it will be, that is what, if you're facing any sort of discomfort, like on my day one only, when I was feeling this feverish, the symptoms were coming on the second day, I tested myself and it actually came out positive. So
Oh,
yes, so that is what,
I'm
and
just
then we
thinking,
have to
and in total webinar session, I just thinking about that. Uh,
my
how
whole
can we
family got
survive
tested
actually?
positive. Whole family. I have
Oh,
8 people in my family. So each one of us got positive, including my son.
oh.
So yes, but we survived.
So
That is
sad.
what.
But now
No, no,
now
no, not
now
so
it's time
bad
for
because we have that
congratulations.
antibodies now in our body. So we are
Now
safe
you don't
for.
need to vaccinate.
No, no, it's not like that. Whenever it comes, like I'm not in that age slot right now, 45 and above. So whenever that age slot comes, I'll definitely be taking up the vaccines. And it's a myth. Please don't believe that when just people think that vaccination can cause death. This is a total myth. It does not happen. It cannot happen.
Yeah,
Okay,
that's true.
if I
That's
could
true.
take up a class
It
on
builds another
vaccination and
immunity in
immunology.
your body.
Yes, yes, it never
It's,
happens. It cannot happen.
it's,
Yeah,
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 that's true.
yeah.
Thank you so much,
Thank you so
today's
much.
speaker. See you again in
Thank
another
you so much.
any topics, another session. So thank
Sure,
you
sure.
so much. Thank you.
Thanks for giving us this platform. Definitely. Thank you so much.
Yeah.
It's my
Thank you so
pleasure.
much.
It's my pleasure. Dear audience, but certification system is always same. You have to browse www.edu.igp.com, and if you are new, then you have to create your own account. If you are already, if you have account, then just log in. For today's class code, you don't need any class code for certification. Why, Salma, you're laughing on behind of the screen? And you can always check, verify your certificate from anywhere. And our next webinar was on held on teaching, sorry, teaching pre-calculus, April 11th April at same time. Thank you, and see you again in the next session. Thank you so much. And I already shared the certification link into comment box and already pinned into Facebook live post. So you can also check it there, and if you are not able to check or click, then you can log in directly from your Google www.eduigp.com. Thank you so much. See you again.